Okay, so today is our ninth lecture, and we're going to talk about a new omics technique, which is the chromatin immunoprecipitation, followed by DNA sequencing or ChIP-seq, which will allow us to identify DNA motifs or DNA sequence patterns where proteins or other molecules can recognize and bind to the DNA for kind of gene regulation or other effects. So today's agenda, we're going to talk a little bit about epigenetics. You probably learned this in biology class already, but we'll kind of make a quick recap of what it is and why we want to study it. And the technique that we're going to use to study some of the epigenetics mechanism, which is the chromatin immunoprecipitation, and then how to analyze the data to identify patterns and then identify DNA motifs. So we are interested in epigenetics because when we want to understand the activities of genes and proteins inside the cells, right? We can monitor gene expression. That will be our next module, transcriptomics. But if we want to understand why a certain gene, why its expression can increase or decrease in the disease or in response to treatment, then we want to know kind of which regulatory mechanisms like increase or decrease the expression. And many of those mechanisms occur on the epigenetics level, which is the regulation of gene expression that doesn't involve any change in DNA sequence. You can have mutation, and mutation can change things, but even without mutation, in our body, like, we have different tissues, like different organs, all have the same DNA, all have the same genome, but they have different gene expression. So the regulation or the determination of gene expression doesn't lie in the DNA sequence alone, but in something that modifies the gene further. So some of the major mechanisms that we're going to talk about today, DNA methylation. When we add methylation to certain nucleotide, it can change the state of the folding of the chromatin. It can turn gene on and off. In later lectures, we will talk about techniques that can measure the accessibility of chromatin, whether the chromatin is open or closed. If chromatin is open, then transcription factors and other proteins can bind to the DNA and activate gene expression. If the DNA is closed, like it's packed, then protein cannot access, and you will have reduced gene expression. And another mechanism is the modification of histone, which we'll talk about shortly. So one popular mechanism for regulating gene expression is via transcription factor. So transcription factors are proteins. So this protein can bind to DNA at different locations. It can bind to the promoter of a gene, and it stabilizes RNA polymerase. So with transcription factor bound to the promoter, RNA polymerase can bind more strongly to the DNA, and RNA polymerase can then transcribe the gene. So these are transcription factors that activate the gene expression. But some transcription factor can bind somewhere else along the DNA at position we call repressor. These location of the DNA, when bound with transcription factor, can fold in 3D onto the promoter and block RNA polymerase from accessing the promoter. So transcription factor can also reduce gene expression by blocking RNA polymerase from certain genes. So in today's lecture, we want to understand how we can use omics technology to study the binding, the activity of transcription factor. So that's the first mechanism. The second one, we want to talk about histone and nucleosome. So histones is a family of protein. There are multiple forms. So this histone will form dimer, tetramer, and octamer. So 8 molecules of histone together form a single structure. And this structure, the special property is that DNA in our body, which is very long. Like we have, humans have 3 billion nucleotides on our genome. Like, so these DNA sequence, which are very long, has to be packaged. And these histone, like when they form the octamer, one of the key functions is that DNA strand can wrap around these octamer molecules, about 150 base pair, can wrap around the one octamer of histone. And this unit of histone and DNA is called a nucleosome. So whenever you see these terms, now you understand that, okay, histone are protein which form octamers, and its functional role in our cells is that it helps package the DNA, organize the DNA by allowing the DNA to wrap around or coil around its, the histone to form a nucleosome unit. So why are we interested in histone and nucleosome? That's because histone, if you notice the diagram here, so histone has a 3D structure where the body kind of fall into a certain shape, but it has a free N-terminal tail. So these are amino acids that are not involved in the 3D structure. It's kind of free, and this amino acid can be modified either by acetylation, phosphorylation, methylation, any PTM that you are familiar with from biology class. And the interesting things about the modification of the histone is that whether you add acetylation, phosphorylation, or methylation, it can affect whether the genes that are near these nucleosomes, it can affect the expression level of genes surrounding that location. And to give you some example of the regulatory function of histone modification, if you have a methylation adding of the CH3 group onto the histone family, like histone number 3, H3, and on the lysine at the 4th position, if you have methylation at this position, these can recruit RNA polymerase. So if you have a lot of methylation on the histone number 3 at lysine number 4, it can recruit RNA polymerase, and it can activate the transcription. So you have more gene expression. And when our cells wants to turn off this gene, we have proteins that can remove the methylation. So I'm not going to ask you to remember all these kind of protein name, but just to show the mechanism that in our cells, if we want to turn on a gene, we can methylate the histone tails, a specific position on the histone tail, which can recruit polymerase and activate the transcription. If our cells wants to turn off the gene, we can demethylate it. We can remove the methylation using a different enzyme. And after demethylation, our cells also have another mechanism. We can shift the methylation to a different position. Instead of the lysine at the 4th position, to the lysine in the 9th position. So a different kind of location on the tail. And just this kind of shift from 4th lysine to the 9th lysine, like a very small change. But when the methylation changed to lysine number 9, these signal a different protein, HP1. This protein package the DNA more closely. So it creates a heterochromatin packaging. So you're transforming from methylation of H3K4, which activates the gene expression, to a demethylation and then methylation of a different site, H3K9, which instead of promoting gene expression, it packaged the DNA and closed all the transcription. So now you have a deactivated genes. And when our cells want to reverse this process, we have a different enzyme that can demethylate or remove the methylation from the histone H3K9. So in our cells, we can turn on or turn off genes by just changing where the methylation occurs. So knowing the signals of modification on the histone tails provide a lot of information about whether that area of the genome will be activated or be replaced. We can tell from the histone signature. So we want techniques that can tell us in different regions of the genome if it's methylated and if are the methylation on H3K4 or on H3K9. And this is just one example of the histone modification. There are other modifications of histone that can affect gene expression. Okay. So the techniques that we want to use is the chromatin immunoprecipitation. So as the name implies, so we're going to do something to the chromatin or the chromosome. And we're going to use antibody to precipitate or to pull down certain proteins. And in this case, you can probably guess that the protein that we want to pull down, one of them would be the transcription factors. We want to know where the transcription factor bind to the DNA. And the other one would be the modification of histone. If we can pull down specifically methylation of H3K4, or if we can pull down specifically methylation of H3K9, then we can study the state of the histone in our cells. So with chromatin immunoprecipitation technique, or short I will call it ChIP, ChIP. So the idea is simple. In our cells, we have protein, transcription factor, bind to certain position on the DNA where it can recognize, or we can have histone modification, like methylation, acetylation, in certain part of the genome. So what we want to do is we want to digest the DNA into short strands and then isolate only the strands that are bound by the transcription factor or are modified on the histone. We want to selectively enrich this DNA. We can do this with antibody. If we have antibody that are specific against certain transcription factor or specific against certain histone modification, then we can pull down the DNA and the protein that are bound to the DNA. And we can isolate just these proteins and do sequencing. So at the end of the day, the DNA sequence that we get will be the DNA sequence that are specific to the position where we have the transcription factor or when we have a certain histone modification. So I just want to point out that when you design a chromatin immunoprecipitation experiment, one of the key factors is which antibody to use. You want your antibody to be very specific to the protein or to the histone that you are studying. And some people have analyzed kind of different antibody from multiple kind of companies. Like there are many companies that will sell you the antibody for certain modification of the histone. In this case, I take the catalog from one company. They have the antibody for histone 3, the 4th lysine with 1 methylation, or histone 3, right, the 4th lysine with trimethylation, 3 methylated groups. There are antibodies that are specific to even the number of methylation on the histone tail. But sometimes if you buy antibody from some company, people have found that the specificity of this antibody might not be reproducible. So you have to kind of do a lot of literature review to pick kind of which antibody to see, because that will affect the entire experiment. So when we design ChIP-seq experiment, the other thing is, as we will see in the analysis part, for ChIP-seq we need a control data. And the control in this case is the DNA sample without immunoprecipitation. So we want to know, like, if, because most of you are biologists, so I'm pretty sure you recognize that immunoprecipitation is not perfect. There is no antibody, there is no pull down that can perfectly enrich only certain protein and with no free DNA coming out. You're going to have some free DNA that have no transcription factor, that have no histone modification being in your sample. So what analysis techniques wants you to do is they want to understand if we don't perform any immunoprecipitation, what kind of sequencing data are we getting? So that's our control. So to look at some visual example, if you have your ChIP-seq data, you are going to have a lot of reads, a lot of sequencing data where the transcription factor bind. But in some location, because of the PCR bias or because of the sequencing platform, you're going to have more read at certain location compared to the rest. So you see some kind of I have higher read count here, but it's not as high as here. If we have no control, if you have no control sample at all, you might be wondering, is this my transcription factor binding site, but it's a weak binding site? Or is it just by chance that I get many reads being aggregated here? But if you have your control sample with no immunoprecipitation, and you have the pattern on the bottom here, then you can compare, like you can tell that, oh, these are probably just noises, right? Because even without immunoprecipitation, I'm getting more reads at this genomic position. So with control data, you can clearly say that this is my transcription factor binding site, and these are not. You can tell from the data. So in the experimental design, you're going to have to spend more money to prepare more sample and to sequence more, because you need to sequence the control as well, not just your ChIP-seq sample. But the good news is, for ChIP-seq, where we only need to identify the location of the peak, like where we have many leads, you don't really need pair-end sequencing. We mentioned before that to understand, to map the sequencing read to the genome, like if you have pair-end data, you have forward read and you have the reverse read, you can map the data more confidently. But for ChIP-seq, where the goal is to identify where the reads accumulate, the algorithm that we have is good enough even if you do single-end sequencing. You sequence only the forward part. So you can reduce your sequencing cost by half by you don't have to sequence both ends, you sequence just one end. But you have to do sequencing twice because you have your control sample. So it kind of comes back, cancel out. You have to spend the same budget as a regular sequencing experiment. So when we want to analyze ChIP-seq data, so it's also sequencing data. But the goal is now different. We want to identify the peak where we have many reads, which tells us that this is probably where transcription factor bind, or this is where the histone modification occurs. So we want to analyze the data. So what are the key steps? Up to this point, I think we have seen many pipelines already, like for variant calling, for metagenomics. And we are going to see similar pipeline throughout. So starting from quality control of the reads of the sequencing data, like with FastQC, we align the read to the genome, we cover this. But what's new here is that we need to call where the peaks are, like where we have a lot of reads. And then we have to assess the quality of the peak that we identify. And then we are going to do two things. First, we want to understand if our peaks, like the where transcription factor bind or where histone modification occur, does it associate with any gene function? Like, are the nearby genes being involved in certain pathway? Because if transcription factor bind at certain location, we want to assume that it's going to activate genes near that location. So we want to understand in our disease or in our treatment sample, when transcription factor bind certain location, or when histone modification occur at certain location, do they try to activate or do they try to replace certain function of the gene, certain gene pathway, for example. The second thing we want to analyze is for transcription factor, if it binds to certain location on the DNA, what kind of DNA pattern, what kind of DNA motif does the transcription factor recognize? Like, does it bind to the Tata box, like TAA, TA, or other pattern? We want to understand these two things. So today's lecture, we are going to finish with these two analysis goals. So let's start with the kind of processing first, like with peak calling. So the idea of peak calling, let's start with the example for transcription factor because it's easier. So let's say we have transcription factor binding site being located at this position. When we digest the DNA and we enrich for the transcription factor with the antibody, we are going to get DNA fragment located around this region because this is the molecules where the transcription factor bind. When we sequence the data, let's see from just a single end. On the sense strand, we're going to get reads that map to the left to the 5' location of the binding site. On the antisense DNA, we can get reads that map to the 3' direction of the binding site. So in your data, in your sequencing data, for single end sequencing data, you're going to see two peaks, one being located to the 5' of the binding site, one being located to the 3' of the binding site. And what we are going to expect is that the distance d here between the two peaks, distance d here should reflect the DNA fragment size. Because on each piece of DNA that surrounds the transcription factor binding site, we're going to sequence either the 5' end or the 3' end. So these two peaks, one located upstream, one located downstream, the distance between these two peaks should be equal to the length of the DNA fragment. So this is one piece of information that we can use. But not now. For now, we just want to recognize that from sequencing data, we're going to be able to identify peaks where we have more read count than other regions of the genome. So this will be our algorithm. We can just count where we have more peaks. But as a bioinformatician, we also want to be able to say statistically, is the peak height, is the read count that we are seeing here, is it significant? Are we confident that it's the transcription factor binding site? And we can do that by comparing the read count in the ChIP-seq sample to the control sample. The question is, which statistical model to use? We have two read counts. We want to compare, let's say, 1,000 read count versus 100 read counts. Is it significant? Which model would you use? So when we think about statistical model, for counting the number of reads, the number of reads are integer, 1, 2, 3, 4. One distribution that might come into your mind is the Poisson distribution. So Poisson distribution, the definition is we have a certain event, certain event basically, that can occur within a certain time period or within a certain location. And you have some expectation that if you observe this event over a certain time, like 1 hour, or certain space, like over 100 base pair, you get to see a certain number of read counts. Let's say 10. Lambda here is your expectation. Let's say you expect that within a span of 100 nucleotides on the genome, you expect to see 10 reads. But in your ChIP-seq sample, you see 100 reads. So you see the sequencing, like the event here is the sequencing. You see 100 reads in your ChIP-seq data. That means you observe 100 reads. Your sequencing occur 100 times within this 100 base pair box. But in your control data, like in your control, there's no immunoprecipitation. You have 10 reads. So this is your expectation. If your ChIP-seq is the same as your control, like there is no transcription factor binding. If in your control you see 10 reads, you should expect to see 10 reads in your ChIP-seq. The fact that you see 100 reads, which is much higher, is an evidence that you are probably enriching for transcription factor binding in this area. And to just for your information, this is the probability for Poisson distribution. If you expect 10 reads at a certain position based on the control data, but in fact you see 100, K here is going to be 100, and lambda is going to be 10. So this is how we kind of plug in the numbers into a Poisson. So in the context of ChIP-seq, so let me say this again. ChIP-seq, the Poisson distribution that we are using here, it explains the probability that you will observe K reads in your ChIP-seq sample when in your control you only observe lambda reads. You replace K with 100 and lambda with 10. So your null hypothesis is that there is no peak. The 100 reads that I'm seeing in the ChIP-seq data is just noise. This is your null hypothesis. There is no transcription factor binding at that location. So if there is no peak, if there is no transcription factor at that location, then we expect to see the same number of reads in ChIP-seq sample compared to the control. So we use lambda or 10 reads from the control as our parameter for the Poisson distribution. But in fact, we observe 100 reads, like the K here is 100. So we can calculate the p-value. What's my probability of seeing 100 reads or more in the ChIP-seq data? When in fact, in the control, I only saw 10 reads. I can create the p-value. So if the p-value is low, that means you have a lot of reads, like 100 reads, much higher than 10. So when you have low p-value, then you reject the null hypothesis that there is no peak. And then your conclusion is there's probably transcription factor binding at that location. And this is one of the reasons why we need the control sample. Without the control sample, we cannot model the Poisson distribution because we don't know the expectation. Like we have no idea how many reads we are going to get at a certain genomic position if we don't have the actual data. So the fact that we know that there are maybe 50 reads here, 10 reads here, 5 reads here, is because we sequence the control sample. So that's very important to do statistics. So that's how we call peaks in each ChIP-seq sample. But let's say you have two conditions, not just control, like you can have treatment or untreated condition, or you might have a disease and healthy condition. You want to compare, is transcription factor binding higher with the disease or lower with the disease? So in this case, we're going to perform peak calling on two different conditions. One for the treatment, like maybe you treat the cell with the drug, and the other one you didn't treat it. But both these are immunoprecipitated samples, so these are ChIP-seq samples. Sometimes you might see transcription factor binding in one condition. So maybe your drug or maybe the disease open the chromatin, and then transcription factor can bind. Without the drug, without the disease, the transcription factor cannot access the DNA, so you have no peaks. So sometimes you can see this pattern where your experiment condition create a new transcription factor binding location. Sometimes you see low binding, you see some peaks, but it's not that high in one condition, which become higher in a different condition. So this can be like you might have an upregulation of transcription factor. So you have more molecules of transcription factor. So in one condition, transcription factor only by a certain frequency. But when you have upregulation of transcription factor, you have more binding. So you can have a differential peak height as well. So you see peak in both conditions, but the height are different. So this can suggest that you have a differential expression of transcription factor, for example. Or it might be the same. Like you might see the same peak at the same location with similar height. So this tells you that this transcription factor at this location, the binding at this location, is probably not related to your disease because it's the same regardless of the state. And we're going to talk about how we can compare the height, like quantitatively, like the height of 100 reads versus 200, when we talk about the next model, which is transcriptomics, when we do differential expression analysis. For now, we just want to recognize that peak calling is the first step, like we identify transcription factor binding site. The next step is we want to compare the peak location and the peak height across conditions to understand the change in transcription factor binding and the change in gene regulation. Okay. So I start with transcription factor binding because it's very simple in the sense that protein recognize a specific DNA sequence, specific DNA motif. So the peak is going to be very clear surrounding that DNA pattern. But when we talk about histone modification, when we have let's say a double methylation of histone 3K79, it's not going to be at specific nucleotide position. It's going to occur over a span, maybe 1,000 base pair or 5,000 base pair. You're going to have methylation of histone, of every histone in this area, because this is how epigenetics turn on or off genes as a region, not just specific gene, but an entire region. So you're going to have enrichment of ChIP-seq data, the read count, that are very broad. It's not going to look like single peak. So to compare, CTCF is a DNA binding protein. So your ChIP peak is going to be very narrow. Like you have one peak here, one peak here, one peak here. It's almost a single position. But when we are analyzing histone modification, it's going to occur over a span of DNA. So you can imagine that the algorithms for detecting the peak is going to differ a little bit. There are two ideas that we can apply to handle such broad peaks. So let's imagine what happened. Let's say histones surrounding this area, many histones, are all methylated. So when you do the antibody pull down, you get to get DNA fragment from everywhere in this region. And when you perform single-end sequencing, your reads are going to span a very broad area. So one type of algorithm that we can do is we can try to merge them. Right? If we have two peaks where the start and the end kind of overlap with each other, then maybe we can just merge them into a single peak. We get a broader peak. So this is one idea. But it's pretty subjective, right? How much of an overlap do we merge? So people try to come up with way to score the overlapping nature of these peaks and to make it so that it's automated. We don't have to kind of look or make some judgment. So one idea, we're going to see this idea again in transcriptomics module. But for now, let's consider this idea. So let's say you identify many locations for the histone modification. And we notice that in some region, we have a high frequency of histone modification. Some is very spread out. So the assumption here is that this region and this region, where we have a high density of histone modification peaks, these are probably a single DNA region that are all methylated, for example. So we want to identify this region as a single peak, not as 7 peaks. And we want to identify this region as a single peak, not as 3 peaks. So one idea that we will see again, as I mentioned, is we want to calculate a kind of a cumulative score going from left to right. Let's see if we can make sense of this. Let's follow this graph. This is the cumulative score. Whenever we see a histone peak, we increase the score. Let's say by the read count. If we don't see the histone peak, we reduce the score. When we see the histone peak again, we increase the score. When we don't see the histone peak, we reduce the score. So what happens is, in a sparse region where we don't have many histone peaks, the score will goes down, goes up, goes down, goes up, goes down, goes down. It never accumulates. But when you see a region with high histone peak, high frequency, the score will goes up, goes down a little, goes up again, goes up again, again. You have an accumulation of high score. And when you enter a sparse region again, the score will drop. Up and then down, up and then down. And when you encounter a new high frequency region, the score will accumulate again. So with this idea, we can argue that if we focus on two specific locations, one where the score is lowest and one where the score is highest, we identify these 4 positions. Because of the way we calculate the cumulative score, the lowest point is going to be the start of a high frequency region, and the highest point is going to be the end of the high frequency region. So we can use this algorithm, calculate the cumulative score, and then find the maximum and the minimum, and say that the region that goes from minimum to maximum, this is my peak, this is my one peak, and this is another peak. So this is also another way that people can use to identify histone modification, which are very broad, by calculating these types of scores. So hopefully you see the intuition behind why this kind of cumulative score and then finding the minimal and maximal point help us identify this high frequency region objectively, right? Because we don't have to make any judgment, right? We just calculate the score and then we find the maximum and the minimum, and that already automatically tell us where the peaks are. Okay. So up to this point, we have seen peak calling ideas for both transcription factor and for histone. So what you are getting from ChIP-seq analysis will be this kind of file, which is sometimes called the base format. So base format is very simple. It's a tabular data. The first column is the sequence name. In this case, it's chromosome name. So this says that the first peak is on chromosome 14, and the location of the peak goes from this base pair to this base pair. So that's the location of the peak. And then it might follow by read count or quality score. So the other columns will tell you the confidence of the peak. And it's not fixed, like these numbers, it can change depending on which software you use. The only thing that's fixed are the first three columns. The first three columns of the base file format has to contain the chromosome ID, the start base pair, and the end base pair. So these can designate the region of the genome. So in addition to telling you about the peak of the ChIP-seq data, if you ever do exome sequencing and you want to know so which exons are included in the exome sequencing, each company will also provide you with a base file that says that, okay, in our exome sequencing panel, we sequence chromosome 14 from this position to this position as an exon. So the base format is a general format that can describe any region on the genome, not just a ChIP-seq peak. It can describe any region. And this file you can put in other software to visualize where the peaks are, because it contains these coded key. So there are several ways that we can check the quality of the ChIP-seq result. After we call the peaks, there are several ways that we can check the quality. So the first one, which is abbreviated as RIP, not rest in peace, is the percentage of reads that are in the peak area. So why is this important? Because when we perform ChIP-seq, we are not sequencing the entire genome. By the design of the chromatin immunoprecipitation, we are hopefully sequencing only the region with the protein. So if our sequencing data, when we align to the genome, if the reads align outside of the peak, that means we have a lot of free DNA that we didn't want to get. Every read that we got, if we do a perfect chromatin immunoprecipitation experiment, all the reads, all the DNA that we get should map to the peaks. So the percent of read in peaks here can tell us how much sequencing data correspond to the design of our chromatin immunoprecipitation. The second and the third metrics here, SSD and RIBL. So the SSD, I forgot what it stands for, but the idea is, so imagine similar as before, in a perfect ChIP-seq experiment, all the reads should be in the peak area because we don't sequence DNA from other region. Okay. So the idea is, if we expect that our peak should be concentrated in the peak area, if we calculate the coverage of the genome, it should be uneven. Right? In the peaks, we should have high coverage. Outside of the peak, we should have low coverage. So SSD here calculate the variance of the coverage of the genome. If we see a uniform coverage, that means our chromatin immunoprecipitation experiment is very bad because we don't enrich particular area. But if we see high coverage, low coverage, high coverage, low coverage, that means we are enriching certain region of the genome. So this score, if it's high, it means that our sequencing data are focusing on certain genome region, which might be a good sign that we are doing chromatin immunoprecipitation for specific region. RIBL, BL here means a blacklisted region. In this case, what it's trying to do is there are certain regions on the DNA where we can have high read count. So this is the nature of the DNA sequence, the nature of the experiment. We're going to have high read count in certain regions, such as microsatellite region, mobile element, repeat region or ribosomal DNA. We're going to have more read count in this area just by the sequence, the distribution of the sequence in the nucleotide in that region. So we want to know if our sequencing data, are they biased toward those regions or are they more balanced? So this RIBL percentage can tell us whether our sequencing data, are they in these kind of spatial region or outside. So it can tell us about the quality of the chromatin immunoprecipitation again. And the last one here, relative CC here. So recall that when we talk about the calling the peak for transcription factor binding, let me go back a little here. Okay. We say that because transcription factor bind at a certain specific kind of nucleotide pattern, when we look at the peaks, we're going to have two peaks, one upstream of the binding site, one downstream. And this distance between the two peaks should reflect the DNA fragment size. And if your library, like when you prepare the DNA, you are preparing the DNA so that all the fragments should have similar size. So what that implies is that in your ChIP-seq data for transcription factor, the two peaks that you found, upstream and downstream, the separation between them, the distance d here, should be very stable because your fragment size is very stable. So this relative CC score check the consistency of your DNA fragment size, the distance between the two peaks. If you have a consistent separation, that means that your two peaks are capturing the DNA fragment. But if your two peaks are sometimes further apart, sometimes very close, sometimes very far, like that means you have something unexpected in your ChIP-seq data or in your library preparation. So this relative CC, if it's high, it tells you that your data looks normal. Your DNA fragment size are the same. The separation between the two peaks for transcription factor binding are very stable. Okay. So this is one way to kind of computationally check the quality of the ChIP-seq result. The other would be to visualize it. We mentioned quickly about integrated genomics viewer before. So this can take in alignment data or the base file that contain all the ChIP-seq peaks. And you can visualize where the peaks are on the genome. So you can see if it occurs near the gene that you want to study or not. But this can still be difficult, like the entire genome is very big. If you have to examine all the ChIP-seq, you're going to have to spend a lot of time. Another way that people use to visualize ChIP-seq data is, let's say we want to study transcription factor. We know that some transcription factor likes to bind in the promoter region, or they like to bind upstream, like 5' from the gene, from the transcription start site of the gene. So instead of visualizing all the ChIP-seq peak kind of all over the DNA and all over the genome, what about if we normalize the location of all the ChIP-seq peak relative to the transcription start site? So in this kind of visualization, which you will commonly see in many in ChIP-seq study, the x-axis is no longer the genomic coordinate, but it's going to be a relative position from the nearest transcription start site, whether our peak is located at the transcription start site, which would be 0, inside the gene body, which would be positive, or upstream from the gene body, which will be negative. Negative means upstream from the gene. Positive means going to the gene body. 0 means the transcription start site for that gene. So people like to summarize the data this way because we can see if our transcription factor or histone modification or RNA polymerase, does it accumulate near the gene? Because we cannot interpret histone modification that are very far from the gene because we don't know if it will affect the gene or not. But if it occurs like within 500 base pair in front of the gene or inside the gene body, then we can interpret that I have an enrichment of histone H3K4 trimethylation, both 500 base pair upstream and inside the gene body for the blue sample. It might be your disease condition, it might be your treated condition. And your control might be the black line here. So where we don't see many peaks kind of before or inside the gene. So we can compare that across the gene, I see enrichment of methylation or I see increased polymerase binding in the blue condition compared to the red and the black condition. So this allows you to kind of summarize data from many genes together on the same graph instead of kind of viewing the specific position on the genome. And next, once we identify all the peaks, we know where the modification of the histone occur, we know where the transcription binding can occur. Next, we want to understand, do these peaks affecting certain pathways, certain gene function? Are the epigenetic signal that we are seeing here, are they trying to turn on or are they trying to turn off certain gene function, certain pathways? So we need to annotate. For each peak, can we annotate, can we say whether this peak is regulating which gene, is regulating which function? So without any other information, like the best that we can do is we can say that, okay, if I see a peak here for transcription factor or for histone modification, and I see a gene next to it, I'm going to assume that this transcription factor binding or this histone modification is going to be regulating this gene because it's the nearest to the peak. So that's the best we can do. With no other information, we can only assign peaks to the nearest genes. Or maybe to multiple genes. Maybe this transcription factor binding can activate all 5 genes at once because the DNA can fold onto the promoter of different genes. Okay. And then we can look at the genes that are linked to the ChIP-seq peak. If the gene has certain functional annotation, we're going to assume that this transcription factor by the histone modification is going to activate or replace that gene function. So let's say we can annotate the function of all the gene and we can link the gene to the ChIP-seq peak. So the next question we want to ask is, if I see more transcription factor binding at this position, more transcription factor binding, are they activating certain function, in particular, the enrichment of function? So we want to know if the genes that are located near our ChIP-seq peaks, are they enriched with certain function or with certain pathway? We can talk about this again in the transcriptomics module, but now I'm going to introduce some of the functional enrichment analysis idea. The first one is called hypergeometric distribution. So the name might sound very scary, hypergeometric. But the idea is not too difficult. So let's say we have a null hypothesis that the ChIP-seq peak that we identify, it doesn't regulate any kinase. It doesn't regulate kinase in particular. So let's say we want to say that the treatment that we are putting into the cells, we see some change in epigenetics. And the null hypothesis is those change in epigenetics is not related to kinases. It doesn't upregulate or downregulate kinases. So this is our null hypothesis. But with our data, let's say we have 20,000 genes in the genome. We know that some of them are kinase. If we have the gene annotation, we know some of them are kinase. We have certain ChIP-seq peak. We identify from the ChIP-seq data. Some of the peak is going to be near kinase gene, either by chance or because the epigenetic is regulating the kinase. But we don't know yet. We just observe the data. That let's say out of 1,000 ChIP-seq peak, we see 100 being near kinase. So what we want to interpret is what is the probability that we see, let's say, 100 peaks out of 1,000 peaks being near kinase genes by chance, if the experiment that we are working on, that we are analyzing, have no relation to kinase. What's the probability that I will see this by chance, the experiment? What's the probability that I'm going to observe this experiment by chance? Given some information that we know that in the organisms, like human, there are 20,000 genes, and we know that some of them are kinase. Given this information, what's the probability that I'm going to get this experimental data? And this can be described with hypergeometric distribution. I'm not going to explain how it's derived, but if you're curious, so this formula is K, the top number, and the bottom number here with the big parenthesis. This is a combinatorial symbol. This means that you have a capital K object, let's say capital K gene, and you are selecting some of them, small k of them, randomly. How many ways can you do that? So for example, if you have 6 genes, and you want to select 2, so you have a 6, choose 2. You can calculate how many ways you can select 2 genes from a collection of 6 genes. And the idea behind hypergeometric distribution is the way that we can observe this experimental data, like where we see a certain ChIP-seq peak, and some of them being kinase. The number of ways that we can see this observation is we need to pick some kinase out of this gray box, certain number of kinase. We have to pick some of them to be near the ChIP-seq peak. And we need to select the rest of the oval here, the rest of the ChIP-seq peak. We need to select them from the non-kinase gene. And then we divide it by all the possibilities. So the hypergeometric distribution gives us the probability that we are observing this experimental result. So from the probability, we can calculate the p-value. The p-value is just the sum of the probability. So if we get low p-value from hypergeometric distribution, we're going to reject the null hypothesis. And the null hypothesis is there is no association between ChIP-seq peak and kinase. So if we see low p-value, we're going to reject that null hypothesis, and we're going to conclude that, maybe the experiment, the ChIP-seq peak that we perform, the epigenetic change that we are observing, maybe they are specifically regulating kinase gene. So this is how we use the hypergeometric distribution to tell if we have some association between the location of the ChIP-seq peak against certain gene function. You can change kinase to any gene function, and this analysis will remain the same. If you change kinase to, let's say, mitochondrial gene, you have a different number of mitochondrial gene, and some peak will be near mitochondrial gene if you analyze the mitochondrial DNA as well. So you can change this function to, let's say, translation elongation. You have a different number of translation elongation gene, and you have different number of peaks from the ChIP-seq that are located near those genes. So your capital K and small k will change based on which gene function you are focusing. But the analysis will stay the same. You just plug in the number and you get the p-value. So let's see a more concrete example for ChIP-seq data. So we have ChIP-seq peak, and then we link them to certain genes that link to the transcription start site. And some gene in green, these genes have a certain function. So let's say I'm changing to cholesterol metabolism here. So out of 20,000 genes, we have 200 that are linked to cholesterol metabolism. And you have, let's say, 1,000 ChIP-seq peaks. 700 of them are located within a kilobase of some gene. I'm going to ignore 300 peaks that are very far from every gene because we cannot interpret them. We're going to focus on the peaks that are located near some gene. And out of 700, let's say 100 of them are near genes linked to cholesterol metabolism. So we know that in our body there are 200 genes. The 700 peaks, 100 of them are near those genes. So you can have some expectation. If in the 20,000 genes, you have 200 being cholesterol metabolism, that means that you have only 1% of the gene being cholesterol metabolism. But in your ChIP-seq data, you have 700 peaks, and you have 100 being linked to cholesterol gene, like near cholesterol gene. It's 1 in 7. This is 1 in 7. This is 1 in 100. So you can imagine that you have a big enrichment. You expect to see 7 peaks. If your ChIP-seq, if your epigenetic signal is not related to cholesterol metabolism, by chance, you should see this many peaks next to cholesterol metabolism genes. Only 7. But in your data, you see 100. So you can calculate kind of roughly that you have a 14-fold enrichment. Instead of 7, you see 14 times higher than 7. And this can be translated to p-value using the hypergeometric distribution. Another way, I just want to show you different way that we can tackle the same question. The other way that we can analyze the enrichment of the function of genes near ChIP-seq peak is via a different distribution called binomial distribution. So this one might be more familiar to you. So binomial distribution describe what's the probability that I'm going to observe some kind of event that will occur with probability p. So event doesn't have to occur, right? It can occur with some probability. Sometimes you see it, sometimes you don't. You're going to observe this n times. You're going to look at it n times. And you see this happen k times. So this is the probability. Out of n times, let's say 100 times, you're going to make 100 experiments. You're observing a certain event. And you see k times. So out of 100 times, you see k times. And for each of those k times, the probability you're seeing it is p. So we have p time itself, k times, to p to the k. And the rest is going to be 1 minus p. The probability that you don't see that event, to the power of the rest of your observation, which is n minus k. How can we use this for ChIP-seq? How can we link what type event, what type probability, what type trial? How does it link to ChIP-seq? So the idea is, instead of annotating ChIP-seq peak by linking it to genes, we can do the opposite. We can start from the genes on the genome. So each gene has different function annotation. And each gene, because of their position, its position on the genome, the region that can regulate it, the epigenetic signal that can regulate each gene, is going to span a certain base pair length surrounding that gene. So you can imagine that, let's say if you focus on cholesterol metabolism again, and these are the genes with green colors in here. So these 3 genes are cholesterol metabolism genes. So the epigenetic signals that can regulate cholesterol metabolism, we're going to assume that it's going to span these green areas that are near cholesterol metabolism gene. So what we can calculate from this type of annotation, where we work backward from the gene, is we know which fraction of the genome are going to regulate cholesterol metabolism. The length, the combined length of these green regions compared to the entire genome, this is a fraction of the genome that are regulating cholesterol metabolism. At least this is our estimation. We estimate this fraction. So let's say we estimate that cholesterol metabolism genes are linked to 0.0001 of the genome in terms of base pair. And let's say we have again 1,000 ChIP-seq peaks. So some peaks are going to fall inside the green area, some peaks are going to fall outside. So now we can use binomial distribution in the sense that Each observation that we are making is the ChIP-seq peak. Imagine if you are playing darts, you are throwing your dart onto the board. If your dart hit an empty region, you have a failure, you don't get the score. If your dart hits the green region, then you get a score. You hit a region linked to cholesterol metabolism. So what's your parameter in the binomial distribution? You are throwing 1,000 darts, 1,000 ChIP-seq peak. You're throwing 1,000 darts. And you see 50 of them falling inside the green region. So this is your event, falling inside the green region. Happened 50 times. So out of 1,000, you see 50 events. It happened 50 times. Then what's the probability? What's the probability that you will throw a dart, throw a ChIP-seq peak onto a genome, and you hit the green region? It's this fraction. Because let's say you throw your ChIP-seq peak, it can occur anywhere in the genome. So the probability that if you hit the green region, which is linked to the cholesterol metabolism, is a fraction of the genome annotated with the green region, which is 0.0001. So you have some expectation. If you throw in 1,000 darts, and each dart has a 0.001 chance of hitting cholesterol metabolism, you expect less than one peak, less than one dart to hit that region, because this time together is still less than one. But the fact that you see 50, this means that you have a very large enrichment of your peak toward the cholesterol metabolism. And you can quantify the probability using the binomial distribution. At least you don't have to calculate this yourself. There are tools that will calculate the probability for you. But this is the idea, how they can derive different p-values for your ChIP-seq associating with specific gene function. At least we know two ways of doing it. So these techniques have very important limitations. The first assumption that we made, which is very hand-waving, is that epigenetic signal, transcription factor binding, histone modification, it affects the near gene, the nearest gene. But we know that DNA can fold, so epigenetic signal can affect gene very far away from the peak location, 10,000 base pair, 50,000 base pair away. So the assumption that peaks are linked to nearby genes might not even be true. But that's the best thing we can do without any other information. So hopefully in the near future, once we know more about how DNA folds, we know the actual 3D structure of the chromosome, we can tell better if an epigenetic signal at certain location, will it fold to another location? And we need other experiments to understand this. But for now, without kind of clear information on the 3D structure of the DNA, we can only assume that the epigenetic signal will affect nearby genes. That's our best guess. Another limitation is change in epigenetics alone doesn't make much sense in terms of the mechanism, in terms of biology. Let's say we see a change in transcription factor binding. If we cannot confirm that the gene is also upregulated, the transcription factor binding might not change anything. Or if we see, so what we want to do with ChIP-seq data, most of the time, is we need to interpret it together with other omics data. So let's say we interpret ChIP-seq data together with transcriptomics. We see increase or decrease in gene expression. We want to know if that kind of increase or decrease, does it occur because of transcription factor binding, also increase or decrease, or does it coincide with the change in histone marker? If it's not, then the mechanism might be something else. But if yes, then we might have more confidence that say, our gene is activated because of the transcription factor binding. So if we want to regulate this gene, we might want to replace or activate the transcription factor. Or we might need to modify the histone marker, the histone modification. When we see mutation from genome sequencing data, we might want to know if the mutation does it disrupt epigenetic signal. When we have mutation, if it occurs in the transcription factor binding site, does it affect the actual binding? Or is the binding kind of robust to the mutation? So we need to see transcription factor binding data together with mutation to see if the mutation affected. So ChIP-seq is usually interpreted together with other omics, not by itself, in order to understand the mechanisms of gene regulation. Okay, so up to this point, we understand how to call peaks from ChIP-seq data and how we can annotate the function to the peaks. Next, we want to do the second thing, which is to identify if our protein, our transcription factor, does it bind to specific DNA pattern. We know the location from ChIP-seq, but we don't yet know the DNA pattern that it binds to. So what we want to see are something like this. For each protein, does it recognize a GGGA? Does it recognize a GTAACA? If we know this pattern, then we can interpret if a mutation occurs on the binding site. If it changes A to T in this position, it might change the transcription factor binding, because FOXD1 wants to bind motif with A at the fourth position. But if we are studying, let's say IRF2, and there is a mutation from A to T at this position, then we can interpret that this might not change transcription factor binding at all, because this position can be A or T. The protein can bind to both. It's not like this position where it must be T, must be A, must be C, for example. So knowing the motif, knowing the pattern that the transcription factor binding recognize, will allow us to interpret impact of mutation and other information. And I want to mention that the concept of DNA motif is very similar to when we study protein blast, the position-specific scoring metrics, where we don't really know about the protein domain amino acid pattern. And we want to analyze the data so that we know the first position of the domain, which amino acid are dominant. So this is the same concept, but changed from amino acid data to nucleotide data. Okay. So what is the assumption that we're going to use? Let's say we focus on a certain protein, and we see ChIP-seq peak at many locations. Our assumption is that this protein recognizes a specific pattern. And this specific pattern should occur in every peak, because if the transcription factor can bind to all these locations, if it really recognizes this specific motif, you should be able to see this motif everywhere that you see the peak. So what we want is an algorithm where we are given the algorithm, the DNA sequence from these peaks. And we are going to ask the algorithm, can you identify a common DNA pattern that occurs in every sequence? And that would be our binding motif. So there are many ways to go about that, but to not make it too complex, we're going to look at the idea of the algorithm. So the idea which I want you to imagine, we have many sequences from different peaks, each line coming from different peaks. The assumption is, if we have the right motif, we should be able to see it in every sequence. That's our assumption, because the transcription factor can bind here. But in the beginning, we have no idea. But I want you to imagine that if you start with a guess, if someone gives you this pattern out of thin air, if someone gives you this pattern, what you can do with what we already know from sequence alignment, we can find the best match to this sequence in every read, in every sequence. We can find, let me use motif. If I give you this motif, like AATCAGTT, you can do sequence alignment. You can find the best match to AATCAGTT in each sequence. And once you identify the best match, you can combine them together to create the position-specific frequency. The first position might always be A, but the second position, there's sometimes more T. The third position is not specific. The fourth position is always C. Once you are able to identify the best match in every sequence, you can combine them to generate this kind of frequency table. And once you have this frequency table, which is more detailed than the original motif, now you have the frequency data. You can go back to the sequence and you can search again. Can I find a better match to this profile in my data? And you can update your profile. Now you might be able to recognize that the fifth position, I saw more A than G before, but now that I know the frequency, it's actually more G than A if I try to find. And the sixth position sometimes contains a C, not always G. So as we update the profiles, we can find better and better match. And our profile, the motif profile here, will converge to a final solution. And this is the algorithm that they use for ChIP-seq, if you recall. So ChIP-seq, we assume that our protein has a certain domain, and the domain has a certain profile of new amino acid, but we don't know. So with ChIP-seq, we put in a single protein, and BLAST search our protein against all other proteins and find the best match. And once we find the best match, we align them together to create this kind of a frequency table, frequency profile. And then we use the frequency profile to search to get a better match. And then this repeat until we arrive at the final amino acid frequency profile that hopefully match the domain, the protein domain evolution. In the case of DNA motif, we want to identify a common pattern that occurs in every sequence, starting from something we have no idea about. And then hopefully, as we search more and more, we find the frequency table more and more, we should converge to a certain answer. So this is the idea. We just repeat the searching and refining the frequency table over and over. But some of you might be able to imagine that this has some severe problem. If you guess something that is completely incorrect, like if your first guess is really bad, if you guess like all A, right, it's not going to match anything in particular. So the key of how this algorithm works depends very much on the first guess. And I'm going to use this opportunity to introduce you another kind of thinking in bioinformatics algorithm. These kind of algorithms where we need to sample some guess, right? We need to make some guess. The final answer depends on the initial. There are two ways that we can solve this problem. First, we can make the first guess smarter. Because we already know, like, that the motif has to be common in all sequence, why don't we do some sequence alignment beforehand? Why don't we do multiple sequence alignment, or do pairwise local alignment? And any DNA pattern that match, like, across this sequence should be a good guess, because those are patterns that appear in many sequences. The motif should be some of those. So you can guess more smartly. You can do sequence alignment beforehand. The other way, you can do it a brute force way. Who's to say that you have to guess only once? You can guess 1,000,000 times, and you run this algorithm 1,000,000 times. And hopefully one of the million, you're going to get a good first guess that gives you the best result. So a brute force way is you try multiple guesses, and you select the best final result. And the actual algorithm that we are using nowadays is a mix between the two. So we're going to start with some smart guess, but because the smart guess, like, sometimes it limits our initial condition. Because the smart guess, if the correct answer is different from any smart guesses, then we never see the answer, because we always rely on sequence alignment. So people want to also try brute force. We want to try something totally wild and see if it gives us a better result. So let me give you some diagram to illustrate this idea. So let's say the x-axis here, the x-axis is the space of DNA motif. So different position on the x-axis is different DNA pattern. The y-axis is how well that motif match to the sequence in our peak. The lower the better. So the idea is we want to sample the initial guess in a diverse fashion. All these yellows would be our initial guess. We want to generate many different initial guess because we want to make sure that we cover all the possible DNA motifs. And for each initial guess, we're going to let the algorithms, the optimization of the nucleotide frequency profile, we're going to let the algorithm run until it converge to a certain motif. We want to start from many different yellow motifs as an initial guess and run the algorithm multiple times. Each time you're going to converge to one motif, which might be different across runs. But we can score. If we can score for each motif, if we can score how well it matches to our ChIP-seq data, then we can pick the best one. And this idea of trying to have a diverse initial condition and then let the algorithm run to completion to get multiple final answer and then evaluate which one is the best. This is a general approach that we will see again in any physical simulation algorithms and also in AI. This is the way that we build simulation and machine learning model. Okay, but for now, this is the first instance that we see this kind of sampling and then optimization being combined into a single algorithm. Okay. So just to tell you, if you ever have to do this kind of motif analysis, there are many tools, but one of them that you can use is the meme suite. So this is a collection of tools. You can see multiple of them here. So they have tools for identifying motifs from your DNA sequence. So the algorithm that we just mentioned. They also have the algorithm. If you have a sequence, if you have a motif, let's say you know from other papers that this is the binding motif for the transcription factor. You want to search for that motif in your sequence. You don't need to find a new motif. You already know it. But you want to know if this motif occurs in your sequence. They also have algorithm for that. And if you want to test if your DNA sequence, like your ChIP-seq peak, when you identify some motif, some DNA pattern, you want to also be able to say statistically, does my DNA sequence or my ChIP-seq peak, do they enrich for these motifs? Are these the transcription factor binding site in the genome? Are they in my peaks? Or are they everywhere in the genome? We want to see if our ChIP-seq peak cover the majority of the transcription factor binding site, or there are also other sites outside of the peaks in the genome. So we want to test comparing our DNA sequence from the peak area to genomic background. Can we see the same motif in the background? If yes, then maybe the motif that we are seeing might not be actual transcription factor binding, right? Because transcription factor binding somehow doesn't bind to other areas in the genome that have the same motif. Okay. So we're going to talk about these algorithms one by one. So the first, if we have a motif, if someone gives you if a paper gives you this motif for a protein, and you have your sequencing read for your ChIP-seq data, how can you score? How can you say that does my sequencing data contain this pattern or not? So the simplest thing that you can do, because you know the motif, so the motif is a property profile of which nucleotide that you're going to observe at each position. So for example, the first position here, where there is almost nothing, right? This is because all the 4 nucleotides can occur with almost the same frequency. Out of 47 samples, they see 15A, 15C, 8G, and 9T. So this is why there is no pattern for the first position. For the second one, there are 20C and 20T, and only 4A and 3G. So you see C and T being similarly dominant. For the third, fourth, fifth, and sixth, these are pure A, G, A, C, G, T. So our 47 sequence that they have in the database, all of them contain an A at the third position, C at the fourth position, G at the fifth position, T at the sixth. So you have this probability. So let's say you have a new sequence like this. The chance that you will see A, C, 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 A, C, G, T, A, G, C, given that it comes from this motif, it's going to correspond to what's the chance that I see a C in the first position. It's going to be 15 over 47 because from the data I have 15C at this position. What about the probability of seeing the second C? It will be 20 divided by 47. So that's the probability. What's the chance of seeing A, C, G, T? Is 1 because it's all A, all C, all G, all T. What's the chance of seeing A here? Oh, it's very rare. It's only 3 out of 47. So you can score your sequence, your motif in your data, compare against this position-specific frequency, and you get this probability. And if you have a pattern that better match to this, so I intentionally make these two nucleotides, A and G, which doesn't really match, against G and T, which match well. The score is going to be different, clearly different, 30 times higher. But because these probability scores are very small, these are difficult for humans to visualize. So people usually turn this into what's called a log odd. So a log odd, if you haven't seen before, is just a logarithm of this probability divided by 1 minus that probability. So it's called log odd because this is the odd ratio between observing this pattern versus not observing that pattern. So it's P over 1 minus P. And we take the log. So that's why it's called the log odd ratio. And it will turn the probability into numbers that are easier for human eyes. So 0.0014 becomes minus 2.85. 0.031 becomes minus 1.49. But the comparison is still the same. We want to prefer sequence with higher probability. So we still want to prefer higher log odd. Minus 1 is higher than minus 2. So this is the way that tools can score the presence of motif in your data. So let's say we want to have statistical test. We want to know if our ChIP-seq, if it's enriched for transcription factor binding, these motifs shouldn't appear outside of ChIP-seq. Because if the transcription factor can bind, it should bind at many places, almost everywhere, not only at some location. But this is not true when we take into account the accessibility of the chromatin. Transcription factor cannot bind closed, tightly packed DNA. It can only bind open DNA. Okay, but anyway, let's say we want to test statistically whether the motif, DNA binding motif that we see in our ChIP-seq, is it significant? We can compare by creating a background of random DNA sequence. Let's say we sample random segments from the same genome. What we can compare is, okay, let's say I see 100 motif location in my ChIP-seq. How many do I see in the background? Do I see more? Do I see less? Out of all the peaks that I have, how many of them that I can detect the motif? The motif might not appear in all peaks. Let's compare to the background. If I sample a random genomic segment, how many segments do I see the motif? Hopefully it should be lower. And if I score a certain motif and I get the log odd score, hopefully in my peak, I should get a better log score than if I do it on a random genome segment. Because this shouldn't be containing my DNA binding motif. Unless my DNA motif is wrong. then a random DNA fragment might see it too. So the null hypothesis here is that your DNA sequence from the ChIP-seq data are not associated with the motif. The null hypothesis is, no, the motif that I'm seeing is not linked to my transcription factor. So what our expectation is that we expect to see the same frequency of motif, the same fraction of sequence with motif, and the same distribution of log or score between our data and the background is under the null hypothesis. So you can use your favorite test for number of occurrence. You might use Poisson distribution again. For log or score, which are real number, you might do t-test, like you might do Mann-Whitney U-test. So you are seeing the pattern now. Once we know how to calculate the score, we make the null hypothesis by assuming that our data is no different than the control. And then we can compare the distribution of our scores in our data against the control and try to reject the null hypothesis. So one caution that I want to make when I say random genomic segment, this can have a big effect on whether you see the motif or not. Because if your motif, let's pick some example here. If your motif has a lot of G's, right? And your DNA background, you sample regions with low GC content, you're not going to see this motif because you don't have many G's in your DNA. But in your read, your ChIP-seq, because you are studying this protein, it might be enriched in GC content, high GC content region. So what's the important thing here is the tools that will do this statistical test for you. When they generate the random genomic segment, they're going to try to control for the GC content for the nucleotide distribution so that you don't have any bias between the two data sets, basically. So for example, if you select random DNA segment from the same genome, like, you are probably controlling for the GC content to some extent, but it's not guaranteed. So doing your generation of random genomic segment, sometimes you might have to reject some of them if the GC content is too different from your ChIP-seq data. Or some tools might try to generate random DNA, not sampling from the genome, but they will generate a random piece of DNA by trying to mimic your data as much as possible. And we will see some of these ideas later in other modules as well. Okay, so that's everything that I want to talk about today. So see you next time.